Hello, and welcome at the Novosanis EACR Research Grant webinar. My name is José Thijssen, your moderator for today's webinar. I'm the Senior Marketing Manager at Novosanis, part of OraSure Technology. In the summer of 2021, we opened the application process for the Novosanis EACR grant. The grant was open to EACR members, and we were delighted to have received over 50 high-qualified applications. The grant awards the most impactful proposal in cancer-related research using urine as sample type. Urine has shown to be an emerging non-invasive liquid biopsy. After a detailed review by the experts of the EACR, one proposal was selected as the winner, and there were 3 runners-up. In a series of 2 webinars, winners will present their research. On October 18th, we had our first webinar, where Dr. Anne-Cécile Duc and Ms. Antonia Hansek from GROW University in Nîmes in France presented their research. Early detection of ovarian cancer using aptamers in urine. Their proposal was awarded with a runner-up grant. Now to today's agenda. Today we have 2 presentations. One presentation by Dr. Barbara Guinn, who was the winner of the Novosanis EACR Research Grant. Characterization of urine biomarker for early detection of endometriosis. But we'll start with Koen Beyers, CTO at Novosanis in Belgium. Koen Beyers is appointed as Chief Technology Officer and co-founder of Novosanis. He obtained a master's degree in industrial design and product development at Artesius University College in Antwerp. He is an expert in product innovation, engineering, design optimization, and rapid prototyping. Koen is a member of the Novosanis board and leading the R&D team. The title of his presentation today is Explorative Study on Device Architecture for Defined Ranges of First Void Urine Collection.
Good morning, good afternoon. I really appreciate the opportunity to talk to you in this webinar and to give you more insights in Colipy as a device and explain the device architecture and features to you. This is a picture of me just before the start of this webinar. No, of course it's not. Just like we all breathe, sweat, sneeze, we pee. And peeing is of course not that much of a burden, and it's non-invasive and it's pain-free, or at least it should be. Any idea how often we pee in our lives? Any idea? On average, 200,000 times. So it's been an accessible and interesting self-sampling type to work on. I'm Koen Beyers. I have a design and technology background. I'm a founder of Novosanis back in 2013. And since the acquisition by OraSure Technologies in 2019, I'm CTO and Senior Director of Physical Design Innovation. A lot of work we've been and are presenting touches upon potential clinical benefits of first void urine and the importance of a high quality first fraction. In my short talk, I'll address the research and development philosophy we went through to design and validate the physics and the volume variance of Colipy, our device for first void urine sampling. Depending on application, high throughput compatibility, preservation requirements, home-based sampling needs, or analyte concentrations, the Colipy platform consists of a number of volumetric standards ranging from 4 ml or 10 ml, 20 ml to 40 ml, with and without preservatives. Colipy consists of 2 parts: the collection funnel with pre-mounted floater, as you can see on the left, and a collector tube, as you can see on the right. This is how it works. The tube is attached to the device. The user takes position, sitting on or standing in front of the toilet, and urinates in the funnel. The collector tube will be filled with the first fraction of the urine flow, and upon filling, the floater will rise, close the access to the tube, open the exit towards the toilet. No interruption needed of the urine flow. The user can just continue until he or she is done, detach the tube and recap it, ready to ship or process. I think it's clear that a lot of our research and development was focused on usability. The user should be able to do no more than capping a tube and pee. As I just said, on average, we pee 200,000 times in our life. To step up to peeing into a device doesn't seem too harsh. We almost always pee into something anyway, obviously. And looking at the results for ease of use or sample type reference, ColiP scores really well indeed. To consistently collect and standardize high quality volumetric first void urine samples, a robust architecture is needed. Most IP and technical solutions of the device are based on hydrodynamics and buoyancy concepts. We came up with a floater design that's activating a valve system. The maths of buoyancy are pretty straightforward. You have a liquid with a certain density, you have an object with a certain mass and a certain volume, and then you have gravity, which is like the only constant in the equation. And you can apply that to any floating or sinking object or device. The object, in our case, is a floater valve, and in order to get it floating, we had to try to work on the density of it by designing an air pocket. This picture is a few years old now, but we've gone through quite a number of iterations to get the density and the total weight of the floater as low as possible, a matter of grams. In the end, we've developed two types of floater valves, serving two ranges of first void urine. One type to serve ColiP FE5000 and large volumes respectively, collecting 20 and 40 ml, and a second type of valve to have ColiP small volumes, collecting 4 and 10 ml. The hydrodynamics behind it look like this. This is for 20 and 40 ml collection. So the tube is decapped and attached to the device. You will see now a technical cross-section of the device, where you see the funnel and the mounted floater in down position. The floater geometry has a valve towards the tube and a one towards the outlet. So urine flows in through the funnel and enters the internal tubing in the floater and starts filling the tube. The urine falling in the tube will stir and mix instantly with the UCM preservative, in this case. As the level in the tube rises, buoyancy kicks in and the air pocket pushes the floater up. With this, the valve, the access towards the tube, closes and the outlet access opens. So the remainder of the urine flows directly to the toilet. The system will allow for first void collection of 20 ml for FE5000 variants and 40 ml for the large volumes variant. A different geometry but a similar concept for collection of small volumes urine, 4 to 10 ml, again tube and device. A tube of 4 to 10 ml can be pushed, fitted or screwed on the device. Floater with double valve is down, as you can see again. Urine is flowing in the device and guided through valve 1 backwards into the tube. The urine level raises, floater valve goes up to close the access to the tube, and the excess urine is guided to the toilet. Truly a miniaturized variant with a liquid volume of just 4 ml or possibly less. We can push up the entire floater valve assembly. And as you know, ease of use is key. It's a fully autonomous yet passive device, just banking on physics and gravity. In order to collect volumetric within a specific range for different collection tubes, some of which are as small as 4 ml, some challenges had to be tackled. On average, depending on frequency of urinating, emptying the bladder will typically produce approximately 300 to 500 microliters. Linked to that, volume flow comes into play as well, typically ranging from a mere 4 ml up to 25 ml per second. So having a buoyancy force that I just explained to push up a floater of a few grams, that could now possibly be jeopardized by hundreds of milliliters of urine, weighing hundreds of grams, being poured into the device towards the floater. We had to bank on computational fluid dynamics to calculate and optimize the hydrodynamic behavior forces and fluid dynamics. And we designed a funnel size and an outlet that can handle high volume flows. Just to give you a number, even if we have just an average urine flow of 16 ml per second, and we have to add 6.5 ml to a 10 ml tube that has 3.5 ml preservative in it, it would only take 0.4 seconds to fill the tube and to have the floater up in order not to dilute the sample. So you would need a robust design for that. The valve switch over should be instant, and this was verified both in lab and clinical settings with some clear output. For all architectures, the collection is volumetric with narrow ranges, guaranteeing volume on the one hand, but also urine preservative ratio. On the left bar, you see a Colli-Pee 10 ml results, middle bar 20 ml results, and on the right, collection without Colli-Pee, where participants were asked to collect 20 ml of first void urine in the standard urine container. Look at the mean value, but more importantly, the range, the spread. Another result. Here you see 3 different manufacturing batches of Colli-Pee FE5000 for 20 ml first void urine. You see good means, limited deviations, and narrow and neat symmetric curves. Some side notes. The floater valve is unambiguous. There's no risk you'd get midstream, clean catch, or dilution in that tube. And the switch over is instant. It's guaranteed first void. The tube is never gonna overflow. Next to taking care of buoyancy, the air pocket is making a good amount of headspace in the tube. And if your tube is underfilled, the user or patient could pee no more. This again is valve bound. The floater needs its predefined minimum of urine before going up. It is designed like that. A few last things in an overview slide. Just like I mentioned a few minutes ago, it takes less than half of a second to fill the tube. Within that same fraction of a second, the collected urine is mixed with the preservative, assuring instant preservation. The devices are injection molded in polypropylene. The last thing you want is losing content, information, cells, or anything because they're sticking inside the device, rather than ending up in the tube. The polymers used are highly hydrophobic. All urine, all content will glide into the collector tube. All other device parts will be as good as dry after use. In addition, a lot of effort was put into the size of the device and the amount of plastics and polymers used. Colli-Pee weighs about 25 grams. I think we give the perception to the user of throwing away quite a lot of plastic, and that's something we'll have to manage. Because in contrast to that, as a solution, Colli-Pee is a load and go concept, where the collector tubes go onto the trays and carriers as they are. No additional handling, no additional plastic peripherals. Looking at other sampling solutions that weigh, for instance, 15 to 20 grams, 30 to 40 grams are added at the lab site for pipetting, other receptacles, and tubes. We are currently working on the metrics, but it seems like we are saving 25 to 40% in carbon footprint. And as we've been making the device light, compact, and flat, it is suited for home-based sampling and postal delivery. It fits in an envelope, weighs less than 100 grams with all packaging materials included, limiting postal costs to the max. Thank you, and looking forward to your questions.
Thank you, Koen, for giving this dynamic presentation and providing more insight in the design of the Colli-Pee. Now it's my pleasure to introduce today's keynote speaker, Dr. Barbara Guinn. Dr. Barbara Guinn is a reader in biomedical science at the University of Hull in the United Kingdom. She has a bachelor in genetics from the University of Aberystwyth and a PhD from the University of Wales College of Medicine, Cardiff. She worked in the University of Toronto as a postdoctoral fellow and started her own research group at King College in London in 1999. Her group is looking at antigenic targets for the targeted therapy of adult B-cell acute lymphocytic leukemia, colon and ovarian cancer, as well as endometriosis and incidental lung nodules. She has shown that antigens can act as biomarkers for diseases, diagnosis, survival, and subtype, as well as providing new insights into the biology underlying these difficult-to-treat diseases. Dr. Guinn is our EACR Novosanis grant winner, and here you see her together with Mr. Kevin Phillips and Ms. Lehigh Cooksey, who is looking into urine biomarker for endometriosis testing for her PhD research. She will highlight her project, which uses Colli-Pee from Novosanis as a urine collection device to allow for standardization of patient sample collection by ensuring quality and reproducibility of data required to get a urine biomarker into clinical practice. Today, Dr. Barbara Guinn will present the characterization of urine biomarkers for early detection of endometriosis.
Hello, my name is Barbara Guinn. I work at the University of Hull, and I'm going to tell you today about some of the work that we've been doing in my group to identify new urine biomarkers for the earlier detection of endometriosis. This work was started a number of years ago, but because of the EACR Novosanis Award, we've been able to accelerate the work we've been doing, looking at urine biomarkers for endometriosis. My background is as a geneticist, and I did a PhD at the University of Wales College of Medicine, Cardiff, where I was predominantly working on leukemia. In my postdoc, I grew my interest in immunotherapy and was fascinated by the fact that you could identify proteins unique to cancer that we could target with the immune system and lead to the death of diseased cells. What I also noticed was that these proteins were often also valuable as biomarkers, and that we could use them to detect early disease state and also prognosis for patients. And it was due to this that I was led to look at ovarian cancer and more recently endometriosis, two diseases that are very difficult to diagnose and quite difficult to treat. Endometriosis affects 1 in 10 women worldwide, which is 190 million women. It's characterized by the growth of endometrial-like tissue outside the uterus, and it can be found in the abdominal cavity, on the outside of the colon and the ovaries and the womb, but also in the pleural cavity and in the brain in some patients. Patients describe terrible pain associated with endometriosis, most commonly pelvic pain, abnormal periods, pain during and before sex, pain during urination, back pain, and 70% of all women who suffer from infertility also have endometriosis. The average time to diagnosis is 8 years due to the vagueness of symptoms and the heterogeneity that is typical of the disease. But the other problem is that when women talk to each other, or they talk to their GP, family members, their mum, their sisters, we often are greeted with comments about, yes, that's a normal period, and yes, they are unpleasant, aren't they? And still, periods are a bit of a dirty word for conversations. A lot of people still aren't that comfortable with having a conversation about periods. The other problem is that it's so difficult to diagnose. The only way to really diagnose endometriosis is through laparoscopy. And unfortunately, this comes with its own inherent risks. So the best way to diagnose endometriosis would be a non-invasive test. At the moment, endometriosis is expected to cost the UK economy 8.2 billion a year. Most of the cost is in loss of working hours, as well as medical costs associated not only with endometriosis itself, but the problems that it causes in other areas of the body. By finding a urine biomarker for endometriosis, we'd be able to detect it earlier and hopefully give patients the right treatment at an early time point where the treatment would have the best impact. We need a test that's sensitive, that can correctly diagnose a patient who has endometriosis, but also specific, a biomarker that can identify patients who don't have endometriosis and therefore don't need the same treatment. There are a number of endometriosis biomarkers that have been identified in urine, but all of them are lacking in either sensitivity or specificity. And we've recently reviewed the literature just to make sure we didn't miss anything. The benefits of a urine biomarker would be a reduced time to diagnosis, improved quality of life, and a reduced economic cost. And so we're keen to find a urine biomarker for endometriosis. We know that hypoxia plays a role in endometriosis. When the uterine lining comes away from the womb, the level of oxygen in that tissue drops because it's no longer being fed by a blood supply from vessels in the nearby tissue. When this happens, we see a drop in the amount of oxygen in the tissue, leading to hypoxia. And this is also corresponded with an increase in hypoxia-inducible factor. HIF stabilization leads to an increase in expression of a number of proteins, which are known to be involved in different systems that are important for cancer and also for endometriosis. These include angiogenesis, the ability to attract a blood supply, metabolism, the ability to use glucose efficiently, and epithelial mesenchymal transition. And I'll talk about that on the next slide. These dysregulated pathways can lead to endometriosis, and we see the telltale signs of that in a lot of the tissues we examine. So the aims of this study are to investigate the effects of hypoxia on the expression and function of novel proteins, to examine the expression of the novel proteins in patient samples, and to determine the sensitivity and specificity of the proteins that we predict can act as urine biomarkers. The first phase of the work performed in our lab on endometriosis was by Leah Cooksey. She looked at the effects of endometriosis tissue when it was put into different conditions. And so she put THESC, which is a normal epithelial stromal cell line that's been immortalized and is similar to normal endometrium, into either normoxia, which is 21% oxygen, or into hypoxia, and found that the cells proliferated at a higher rate. She did the same experiment with a cell line called 12Z, which was taken from a woman who had endometriosis. And again, she found in normoxia, the cells grew at a slower rate than in hypoxia. Leah performed scratch assays. We take a confluently growing plate of cells and make a scratch along the center of the cells using a pipette tip. And then we look to see how the cells fill in the wound over the coming hours. What we found was that in the low serum conditions that encourage migration rather than proliferation, in normoxia, the cells took longer to fill the gap than in hypoxia. And this was graphed out so you can see how significant that difference was. Leah also did analysis of RNA sequencing data that was available on the web, and showed that there were a bunch of genes that were upregulated in hypoxia compared to normoxia in the cell lines she'd been looking at. What's interesting is the largest number of these genes are involved in cancer. Leah also looked to see which genes were upregulated in hypoxia compared to normoxia, and in cancer as well as in endometriosis, and she found some commonalities. Of note was the presence of MB1, a protein that has been shown to be important in cancer cells and upregulated in hypoxia. She did look at a number of the genes that she'd identified. Using qPCR, Leah looked at 12Z in either normoxia or hypoxia, and then mapped out the fold change in expression of each of the genes in hypoxia, and was able to show that MB1 was upregulated in expression in both 12Z, the endometriotic cell line, as well as THESC, the epithelial stromal cell line. And she was able to show by Western blot that MB1 increased in levels in both 12Z and in THESC in hypoxia. Beta-actin is shown here as a loading control, so you know there's the same amount of protein in each of these wells. And Leah also showed HIF1 expression, so you can see there's less HIF1 in normoxia and it's stabilized in hypoxia. Leah wanted to look to see whether MB1 expression, if it was knocked down, would have any impact on the growth of the cells. And so Leah took her cell lines and incubated them either with scrambled siRNA or with siRNA that targeted MB1. With scrambled with scrambled siRNA, then the cells that were in normoxia proliferated more. But if Leah incubated the cells with the MB1 siRNA that knocks down MB1 expression, she could show that in hypoxia the proliferation rates were decreased. So on the right in the Western blot, you can see that with increasing amounts of siRNA, you see decreasing amounts of MB1 protein, and the beta-actin loading control just showing that we've got the same amount of protein in each lane. Leah then stabilized HIF1 with DFO and was able to show that although HIF1 was stabilized by DFO, it didn't make any changes on the amount of MB1 in the cells. And again, these are the beta-actin controls. And then Hannah Draper has been looking at proteins in the cell lines that we have. So this is H1299, a lung cancer cell line, known to be positive for the expression of MB1, MB2, and MB3, as shown by the dark brown staining in the cytoplasm in the cells. And she could also show that we've got MB1, 2, and 3 expression in 12Z, which is our endometriotic cell line, and also in our epithelial stromal cells. MB1 is found to be overexpressed in multiple cancers. This leads to a more rapid proliferation, an increase in the epithelial mesenchymal transition, and an increase in invasive behaviors. You can see in this picture that from the epithelial state, the cells tend to look more organized, and as they become more mesenchymal in features, then you see changes in cell-cell adhesion, cell individualization, front to back cell polarity, acquisition of cell motility, and basement membrane invasion. We also see a change in the proteins that are expressed from E-cadherin and cytokeratins over to N-cadherin, fibronectin, collagen, snail, and vermentin. And Leah's been looking at these changes in our cell lines in hypoxia. So the second phase of our study has been performed by Hannah Draper. She's been working with Kevin Phillips, Jane Allen, and Keith Cunningham at the BSGE Endometriosis Centre. Since recruitment opened in late January 2022, Hannah's been able to consent and get samples from 83 patients who were thought to have deep endometriosis and were coming into the clinic to have a laparoscopy for treatment, but also to confirm the diagnosis. Currently, Hannah's undertaking a substantial amendment on our ethics so that we can include healthy donor urine samples and blood, and also women with subfertility, and also increase the number of samples that we receive to 500. It sounds like an awful lot, but we think that's the amount we're going to need because this is such a heterogeneous disease. And as you'll see in a few minutes, that when we start cutting down the number of patients we have in any one group, we very quickly get to a small number of patients that we're looking at. So in terms of how we identify patients with symptoms suggestive of endometriosis who are, suffering with the symptoms, maybe they have pelvic pain, heavy or painful periods, cyclical urinary or gastrointestinal symptoms or subfertility, will come into the BSGE endometriosis centre. They'll have a preoperative assessment and hopefully give informed consent. So far, every patient apart from 2 have given informed consent. The endometriosis patients are very happy to help, which is great. Patients will then complete a Worth questionnaire. They'll be asked about their menstrual cycle, what medication they're taking, what other symptoms and diseases they have, and when their last food and drink was. We'll also measure their hip circumference, their BMI, and all of these things help make sure that we understand them as individuals when we're looking at the patient data. And hopefully we'll make future studies even easier. We then obtain a clean catch of urine taken before the patients go in for their operation. This urine is collected using a Colli-P tube shown here on the right. This allows us to collect the first void of urine. During surgery, patients will have ectopic endometriotic deposits removed, and these, any that aren't needed for pathology, will be stored in RNA later. Samples are taken prior to the induction of anaesthetic so that that doesn't interfere with the disease in any way. And 2 independent BSGE accredited endometriosis surgeons will do a visual inspection of the patient and classify the endometriosis as deep, superficial, ovarian or other, with a common occurrence of scar endometriosis, which is when people have had a cesarean and then the endometriosis forms in their scar. There will be detailed operative findings recorded, and some of the tissue will be sent off to pathology. Finally, samples are kept on ice and transported back to our laboratories, where we perform urinalysis, aliquot the samples and freeze them in -80 on the same day. Tissue samples spend the night in the fridge and then are aliquoted and stored in -80 the next day. So I said about the fact that even though we have a large number of patients, the numbers within any one group quickly whittles down. So far we've had 83 participants, 7 are awaiting classification, 2 are awaiting histology, 5 have had their operation cancelled, usually due to a lack of beds. I have 62 patients who've been confirmed as having endometriosis. 32 of them had deep endometriosis, 20 had superficial, 10 had other types of endometriosis, and 14 of these patients were actually found not to have endometriosis, despite having symptoms that could easily be thought to be endometriosis. The age of our patients and our controls are very similar, as are the BMIs, although the ranges are different slightly. But as well as looking at these different types of endometriosis, we've also looked at the different treatments patients have, and at that point we are getting into small numbers. So patient recruitment, we were a little bit behind target until March when the endometriosis centre opened back up post-COVID lockdown, and we've had a little bit of dip in the end of the summer when a lot of the elective theatres were cancelled due to a lack of staff. And then we've had an increasing number of samples collected. We're hoping to get about 100 samples a year, which we're very pleased with. And we're also collaborating with the Liverpool Women's and Children's Hospital. Professor Durrani has offered 150 samples that we can use to confirm our results. So far, we've looked at the expression of MB1, 2 and 3 in urine. We found elevated levels of MB1, MB2 and MB3 in deep endometriosis compared to superficial and symptomatic. We found a close correlation between MB1 and MB2 levels, MB1 and MB3 levels, and MB2 and MB3 levels. We also then took our patients and had a look to see what stage of the menstrual cycle they were in. So menstrual cycle, the bleeding part of a period, day 0 to 5. amenorrhea is women who don't bleed. Secretory is day 14 to 28 after your menses, and proliferative is day 6 to 14. And what we find is elevated levels in the proliferative phase. All of these patients are not on treatment at the moment. Then when we look at patients with hormonal treatment, then we see if patients aren't having hormones, they have elevated levels of MB1, but they have decreased levels of progesterone, decreased levels of MB1 if they're on progesterone, or GnRH analogues, which induce a chemical menopause. And it makes a big difference. We can see very clearly patients who have treatment who don't have the same system to have periods have a lot less MB1 than those that don't have hormones and do have periods. MB1 is also a potential biomarker for adenomyosis. Adenomyosis is where you have endometriosis inside the uterus, and it is invading into the womb itself. Women with adenomyosis are very difficult to diagnose, and so this finding that we can tell the difference between symptomatic controls and patients with adenomyosis is very exciting. So in terms of future directions, we obviously need more patient samples. We would like to see what the impact of preservative is on our detection of MB1, 2 and 3, as this would allow samples to be posted to the lab for testing. We'd like to see whether combinations of MB1, 2 and or 3 increase sensitivity and specificity of our biomarkers. Want to look at healthy controls and see whether we can find MB1, 2 and 3 in the urine. And we also want to see whether MB1, 2 or 3 could act as therapeutic targets. This leaves me to thank the people who've done the work. Leah Cooksey received a University of Hull PhD cluster studentship. She's a young lady with blonde hair, just sat at our Christmas party. Hannah Draper, the lady in green, she is our clinical fellow who's been funded by the Hull University Teaching Hospital, has been working on the patient samples. And of course, I have to thank my colleagues at the Endometriosis Centre who've been so proactive, helping this study come to fruition. We've received funding from EACR Novosanis, which has made a huge difference to what we've been able to do in the last year, particularly around buying a lot of ELISA plates and being able to test women's urine, both for the MB1, 2 and 3, but also for creatinine. We also received some funding from BSGE, which is the British Society for Gynecological Endoscopy. And we can't thank the Endometriosis Support Hull and East Yorkshire ladies enough. These ladies have been so willing to help and support us. It's been really lovely to see. They're incredible when they look after other people with endometriosis going in for surgery. They get a gift bag. They get a guide on what to expect during surgery. They share phone numbers so that you've got someone to ring when you go for surgery and when you come out. They have a Facebook group that's very active so that everyone's got help and support. And at the moment, last count, they had over 500 members and it's growing very quickly. The Endometriosis Support Hull and East Yorkshire are so supportive that they are organising a ball for the 31st of October, 31st of March next year to raise money for research in our group, which I couldn't be more pleased about and can't wait for. So just leaves me to thank you for listening to my talk today. I really appreciate the time you've taken to do so. If you would like to get in touch, you're very welcome to. I'm very happy to take questions. But if you think of a question later, or if you suddenly just fancy having a chat, then please do reach out. And thank you again to EACR Novosanis for this funding. Really do appreciate all the help and support that we've received over the last year or so. It's been great. Thank you.
Hello, good morning and good afternoon to the audience. We are here for the webinar for the EACR Novosanis Research Grant, and we are now at the last section of this webinar where we have a Q&A session. First of all, thank you very much, Dr. Barbara Guinn, for your really interesting presentation on endometriosis and really your really insight in having a great test available that is non-invasive and that can help all these patients that are really struggling to get a diagnosis. And also thank you very much, Koen Beyers, for your presentation on really how does the Colli-P work and also all the design ideas that are behind it and showing us those nice movies here. For the audience, if you want to ask these two excellent presenters a question, please go to the Q&A session and put your question in there, and then we'll address that to the presenters. So while I give the audience some time to put their questions into the Q&A session, I have already here some questions for Dr. Barbara Guinn. Dr. Barbara Guinn, you presented your research approach to test for endometriosis and develop basically your test and find a good biomarker. So the question is, what do GnRH analogs do in terms of treating patients with endometriosis? Could you elaborate on that, please?
Thank you for the question, Jose. Appreciate it. GnRH analogs are gonadotrophin hormone releasing hormone, and the analogs are used to actually block the production of follicular stimulating hormone and luteinizing hormone. Sorry, a bit of a mouthful. And what this does is it basically stops the production of estrogen and puts the women into a premature menopause. And we find that as soon as we stop the periods, then the symptoms of endometriosis begin to dampen. And so the women don't suffer as much. Unfortunately, the downside of this is that when you're on GnRH analogs, you're more at risk for hot flushes, loss of bone marrow, bone mineral density, sleep disturbances, and all those menopause issues that women can suffer from. So we, at the moment, only recommend that women are on GnRH analogs for 6 months after surgery to try and help the endometriosis not reform, and also for up to a year prior to surgery, just to try and get the endometriosis under control.
Thank you very much for your answer. I have another question for you, Dr. Barbara Guinn. You showed a graph with levels of ENBI1 versus state of the menstrual cycle. Do the levels shown in the secretory phase include women with deep and superficial endometriosis, and where do their levels appear in the graph?
Yep, that's a really good question. After I did the presentation, I was preparing it and I suddenly realized I had the same question. And so I've asked Hannah to provide the data, and I'll just, hopefully you can see it now. Can you see a slide?
Yes, that's visible.
Okay, so if I just change it here, all of these bright green icons, these show you women with deep endometriosis. So you can see that even the women with deep endometriosis have less of the MB1 if they're in any other phase other than the proliferative one. So we do see that MB1 levels go down depending on what stage the women are in their menstrual cycle. So probably like some other tests that we do, such as when women go for a smear test, we would say to women, please make sure that you're in that period or that phase of your menstrual cycle after your period's finished and before day 14. So we will have to make sure the test is done at a certain time point in the menstrual cycle.
Okay. It's very good that you have that additional information about when the test should be taken.
Yeah.
And I have here another question for Koen. So Koen, you showed us the Colli-P, and I think it's a great concept. And you have them, as you shown, in large and small volumes available. So if you look at the very small volumes, do these variants have the same volume range accuracy like the large variants? And as a form of a relative conceptual point of view, it feels much easier for large collipees than for small collipees. Could you elaborate on this?
Yeah, that was a challenge indeed. Had we used the same floater concept for the 4 and 10 ml variants as for the 20 or 40 ml, then we had the same absolute deviation indeed. And 2 ml, for instance, 2 ml deviation on 20 ml hurts less than 2 ml on 10 or 4 ml, of course. And that's why we had to make a new miniaturized design. And with that, we scaled tolerance, we scaled the deviation basically. So yes, the small volumes have the same accuracy or quasi the same accuracy than the larger volumes, which is great when you add preservative and have to respect the urine preservative ratio.
Okay, thank you very much for that answer. I now have a question that's on carbon footprint, and I think that's for both of you something you probably look into because if you are developing a test or a product, you really want to make sure that you are using all the materials in an efficient way. So for Koen, you mentioned also in your presentation carbon footprint, and you said it's not negatively affected by using the colipee compared to other methods. Can you elaborate a little bit more what you mean? And also, did you consider by developing and designing the colipee to use biodegradable or recyclable materials?
Well, we are using more plastics at the front end, indeed, during collection, but we are winning that back at the lab site during processing. And as I said, indeed, in my presentation, we probably save more than 25% in use of polymers. Next to that, that's the second part of your question. We have been looking into biodegradables, paper, cardboard, bagasse, and that scouting is continuously going on, I have to say. But we are working in a very regulated industry, and we are to use medical grade materials. So we have to be very careful and build evidence with any material we use. So now we are using virgin PP materials, polypropylene materials.
I think this is something we all think about, how to make sure that we are including carbon footprint. I just see a question coming in for you, Barbara, that's more on the biomarkers you look into. So the question is, could ENBI1, ENBI2, and BNI3 be measured as secondary effects of endometriosis, such as inflammation, rather than the disease process? How specific do you think these markers will be?
We're hoping quite specific. We know that MB1, MB2, and MB3 are involved in the disease process and are not biomarkers of inflammation. So we're quite hopeful that these will also be potential targets for therapy.
Okay. That is good to know that that is also a possibility. What I also find very interesting to know is what are the steps, Dr. Barbara Guinn, that are needed to have the method to present it available for patients in the clinic? Could you explain a little bit more, like what is needed there and how that will evolve, you think?
Yeah. So at the moment, we're at the point where we're validating the technology in the lab. Our work at the moment is to look at more samples just to make sure that the results hold true on a larger cohort. We really want to look at women with endometriosis and also normal donors to see how the phase of menstrual cycle affects the MB levels. And we want to follow women through their menstrual cycle. So maybe not every day, but certainly urine samples from every phase. So we can see how does that affect MB levels. We think that we're going to have to do an ELISA test when this product goes into like a clinical use. And for us to be able to do that, then either we'll need to see women and then send the samples off to a pathology lab, or I don't know, they'll have to come into a specialist centre, you know.
Mhm.
So whatever happens, we'll probably need those samples to be collected into preservative. So the next steps will also look at whether the women can pee into Colli-Pee tubes with preservative. And then this could be potentially a postal offering where women could wee at home into the Colli-Pee tubes and then send the tubes into a pathology lab for testing. So that's some of the things that we'll be doing next. In the new year, our colleagues in Liverpool will be sending us a validation cohort of urine samples. So these are urine samples that they've been collecting over the last year. There'll be about 150 samples that we will test to see, do we get similar results with the Liverpool samples as we've got with our own. So that's the important next steps. And then hopefully we can start to show, develop a prototype, and that will probably be towards the end of next year.
Wow, that sounds really good, aligned with the steps. And Koen, maybe you want to add something because you also showed different preservatives that you have available in the Colli-Pee as well as a postal kit. You're on mute, I think, Koen.
Yeah, I was. Yeah, that was one of the advantages of the miniaturized design. So we could make the funnel a bit smaller, the tubes are smaller. We can make really basically a very thin parcel that would classify as a letter. So for postal or home-based sampling, that's an excellent solution, I think, to receive a letter, to send out a letter. Postal costs alone would go down. We can pack safety bags, absorbing tissues, anything we have to do according to regulations. We can basically pack in a very compact parcel for at-home testing. Thanks to the mini-P design or the very small volume design.
Oh, that is great that we are also providing that possibility for making it easier to have the test that Barbara presented into the clinic. So looking at the time, and I don't see any more questions coming in for the audience at the moment. I wanted to give you, Barbara, if you want, sometime if you want to address something that you were not able to address yet, feel free to take the floor.
Thank you very much. I don't know, you listen to your presentation, you think about what you would like to talk about, and I think for me it's been really good to hear Koen explain how the Colli-Pee system works. I think that our women who've used it have found it very convenient, and I'm amazed that you can pee in a tube, and the way it's designed is so clever that the collection section rises and we only collect the first void, and certainly we've had very good feedback about the system. So it's been a great way to do a first catch and a clean catch, straight into the tube that we then use to aliquot out our urine. So it's made life a lot easier for the women as well as for us.
Okay. So thank you very much for these remarks. Koen, do you want to add something?
Yeah, and I think adding to that, that's why we do it. We can design Colli-Pees in our R&D lab. It's only in true research and in the field that we get the true feedback, that we can measure the true quality of the sample. Next to the usability, I think that's a great thing to have. So I'm really happy with this cooperation we've had and with the results. Thank you so much.
Well, thank you. It's made such a huge difference to the amount of work that we've been able to do and how quickly we've been able to do it. And, it's been fantastic for Hannah as part of her MD to be able to collect so many samples and analyse them.
So thank you both very much for your contribution today to the webinar and your really interesting presentation. And for the audience, I also want to say thank you very much for attending this webinar. This webinar will be available online as a recording, so you can always go back to it or share it with your colleagues. It will be available on the website of Novosanis, and we also will make it available for Barbara Guinn and her team. So for today, thank you very much, and we look forward to seeing you in another webinar or in another live event. Thank you again.
Okay, thank you very much.
Thank you. Bye-bye.
Bye.